I'm Jil Meier, and I've been working on brain stimulation for exactly 1 year now, actually. So quite excited to share all the knowledge that I've gathered with you guys. I have been a postdoc here in Petra Ritter's lab. And today you will see me for the next 3 hours if everything is working, and my screen as well. So yeah, where should we start? Brain stimulation is the topic for today. You have seen the lectures before. We learned about pre-processing, we learned about multi-scale, so zooming into specific brain regions. And then we zoomed into a specific disease, Alzheimer's disease last time, and we really looked what is the cause and what can the cause have on an effect on the modeling with the virtual brain. So, and ontologies and knowledge graphs. So maybe you're still on the homework. Exciting new work there, and this time will be all about stimulation. We will keep it to the brain, so brain stimulation, and this will be the last time where it's all lectures, and next time participants, so you guys, will give presentations. Today we will have the same composition of the course as last time and the time before, so we will switch in between. We will have a lecture where I will tell you everything about surface-based simulations and brain stimulation. I will tell you in a second why we need the surface here. And then we will switch to a hands-on part where we will actually do quite interactively a program with eBrains. So let's see if we seem to be an okay number of participants, so make that work. Let's see how that's going. Then a 10-minute break. And yeah, I will try to keep it really on time, everything, so that you can plan ahead in case, yeah, you want to miss out on something or keep and catch it up later. And then the second lecture part will be about brain stimulation and other neuromodulation techniques. So a bit an outlook and what else could be done and future work, future challenges, and so on. And then there is another hands-on part in the end where we will work with brain stimulations again. But also, I hope we will have time for that in the end, look a bit at the graphical user interface, the GUI of TVB, so that in the end you will have a video of your own first brain stimulation with the virtual brain. Let's see. Okay, I made some learning goals for today, and you should have a link to the slides as well, so you can gather, yeah, Chloe sent it again. Great, thanks, Chloe. So you have access to the slides already. You can see everything, follow everything, and ask questions in the chat if you like, but also, yeah, however you prefer. And I will do my best to answer them directly. The learning goals for today, yeah, this is how I imagine all of you sitting there. We will have 5 learning goals. Goal number 1 is that we will know, hopefully, after this lecture about the choices we have to make, the advantages there are when performing a surface-based simulation. So why do we have to do this? And the second goal would be, I hope after the lecture, you will know why and how brain stimulation is performed nowadays, and give you an overview of previous studies in this field using the virtual brain, but also other stimulation techniques and other literature. Then the fourth goal will be that you will be able, after this lecture, to perform your own simulation with a virtual brain, Python, as well as graphical user interface, where you stimulate a region and you can look yourself how the stimulation, what is it doing to the virtual brain. And the fifth learning goal would be to be able to set up and run a surface-based simulation with a Python interface. Surface-based simulations are about the computationally one of the most expensive simulations you can run with TVB. So we will split that up a bit and not combine it with a surface-based stimulation, but I will come to that in a second. Okay, so the story of today, to give you a small outline, we have to work through first surface-based simulations, understand what that is, and know the benefits of it, because we will need it to then really look in detail into the precise effect of a stimulus in the brain. Then we will have a look, and you will hopefully create those beautiful figures yourself with eBrains, where you can see how a stimulus set into a specific region will propagate over time through the brain. And then in the end, I will put you here for a sec, yeah, that I can see my own slides. Then will be the fun part in the end. So I try to make it lighter towards the end in terms of content. What are the consequences of the stimulus in the real brain? So first we do the virtual brain and then we do the real brain. Can we actually cure diseases with a stimulus? Can we manipulate behavior? What's possible? How much can we, actually alter the brain dynamics with the stimulus? This is where it gets very exciting, I hope. Oh, here we go. Yes, so this is the typical, could be a tweet, right? How it started, how it's going. So this is some historical perspective on brain stimulation. We have probably seen some of those very scary movies. Here, California, 1942, how electroshock therapy is actually has been administered to psychiatric patients in the 1950s and even before here. And this was quite brutal. A lot of people had to help hold that person down, and it wasn't really well known what's happening. They still performed it. And now how it's going nowadays, on the other side, there's Gertrude. So who's Gertrude? It's this happy pig here who has an implant. And this is supposed to be the Fitbit in your skull, as Elon Musk is actually referring to it in August 2020, which is a tiny device, as you can see here, with some little fibers in the end, which is implanted just on the skull of your head. So now you can see Gertrude, the happy pig. And you can see that this little device here by the company of Elon Musk, Neuralink, actually been, trying to find my mouse, here, is a tiny device implanted just on top of your head in the skull, with tiny fibers here, which these fibers, they can actually measure your brain, but also in the future, they're hoping that this tiny device can write to your brain. So actually take up some of the function or alter your brain dynamics. So, and it just a couple of days ago, Elon Musk gave another interview about Neuralink and said that there's somewhere in the lab of Neuralink sitting a happy, very happy monkey with an implant already. And he's playing computer games just based on the chip in his head. And he said he's very happy. So this seems to be not bothering anymore to have an implant in your head. And this, we will see if this will be the future for us with a Fitbit in our skull. So in terms of the virtual brain and everything that you have been learning so far, the individual connectivity, how we can load it, what can we do to the tracts, how can we look at local neural mass model and even go and zoom into a region, find some local parameters. This is where we are here at the local stimulation, and an overview paper of the virtual brain puts this point as optional. So this is what you can learn. You can decide to stay in resting state virtual brain, but you can also go a step further and go towards stimulation. And this is a kind of an extra input you give to the virtual brain. Okay, so this, let's start very easy. These slides you have seen before. It's a general picture of what we're doing when we are modeling the brain with neuroimaging data. And you have learned that these fibers are important and these atlases are very important. And this you have seen before on Micha's slides in the second lecture. This is the surface. And there was something mentioned about, oh, the surface can be very important as well. And finally, we are getting to this part. So what's this surface? It's actually, so you can see tiny little triangles here all over the surface of your brain. And this Micha has already shown you some distributions. He called it local connectivity, kernels, short-range connectivity, but maybe you were wondering what that is. So you will find out in a bit. This is also something you have seen before. So let me place actually everything in here. Yeah, and everything in in perspective in this one global equation of TVB. So this is a differential operator that maps something in time to the next time point. And this is the neural mass model. We have heard different names for that, reduced Wang-Wang or Janssen-Rid. And this is actually the state variable. It can be a firing rate or something that is going through the model and put towards the next time step. And this is very important as well. We have seen we need to optimize this. This is a global coupling scaling factor, with which you can actually alter the structural connectivity matrix. So this is a very important input for TVB, and everything depends on it. And this is also called long-range coupling. Long range because it can be a really long connection sometimes between two regions. And then we have the time delays, which can be very important as well. And here comes the local connectivity, which is a connectivity just between neighbors. So really spatially closed triangles on the surface. I will explain that in a minute. So you have heard that before. And here is the injected input. And this part is where actually the stimulation comes in. In this injected input, we will put in a stimulus. And then the noise can be very important because in reality we have noise. So we need this noise to make our data and our whole approach as realistic as possible. So I have again prepared a small Kahoot quiz for you guys to follow. Oh, my slides are not, oh, it was 5 minutes ago. Okay, my slides are visible. Great. Yes, so I have a small quiz. I will post it here for you guys to play along. So it's just for fun again. Multiple answers can be right, but you can only click one answer. The quiz is again open until 6 p.m. And all you have to do is go to kahoot.it, fill in this game pin, choose a nickname and make it as anonymous as you can. And then everybody is invited to play along, and this quiz will actually follow the different parts of the lecture. So you can only answer these questions so far, and then you have to wait until the lecture goes on, or maybe you know the next answer already, or you look ahead in the slides, however you want to do it, but you have time to follow it as slowly as the lecture, and then at 6 p.m. this will close and we will reveal the winner. Okay, so this is just for you to keep on track and focused. Okay, so surface-based simulations. Why would we do that? What is it and how do we do it? So the motivation is that we have regions before. We always had one region is a node in the network. And now we want to go to a finer scale because regions are often of different sizes, right? They're just shaped in space of different sizes. So we need a finer scale to be as close to reality as we can be. So each vertex of the surface, each little triangle will be considered a node and can be modeled with a neural mass model. So that's the whole idea of the surface-based simulation. You take the surface and instead of every region, on every vertex, you put a neural mass model. And in this way, you include short-range intracortical connections because, of course, if you are spatially close, chances are that your activity with your neighbor is closely related, right? Even though you may be not friends, if they have loud music, like you will hear it, right? Okay, so here are coming the triangles. The surface is made into smaller and smaller and smaller triangles in a resolution of our choice, such that every triangle will be its own neural mass model. Then they will be coupled, of course. The regions will be coupled together by the long-range connectivity or white matter connectivity, which is also called often heterogeneous connectivity, because for different regions, this connectivity is heterogeneous. It will be different for different regions. This is just, so this heterogeneous long-range white matter connectivity is the connectivity you've already seen. This is the one you already know. This is the connectome. This is the matrix we are loading into our models. And then this other dimension that we're adding now is called the local connectivity or short-range connectivity, because it's just between the two triangles next to each other and maybe a few steps further, but not everywhere. It's local, but it's placed on the surface on every triangle of your surface. Everywhere the same. So that's why it's called homogeneous connectivity. So this homogeneous connectivity, which is everywhere the same, is the connectivity that is giving very strong connections in between direct neighbors. And the further you are away from this red part here, in this case, the less your connectivity will be to that part. So it's just adding another dimension of how connected can the brain be, and actually is the brain. And there are these different profiles that you have seen before. So the first profile is the Gaussian profile, which just, the further you are away, this is distance in millimeters, the chances are less and less more likely that you are actually connected to that person. In this case, to that little triangle on the surface. Then another possibility here is a Laplace distribution. So it really goes down very quickly. And those people here being further away, I say people, I mean triangles, are actually having no chances of being connected to this one red triangle. And here, this is a fun distribution. It's called, as it looks, it's called the Mexican hat distribution. That's another choice of local connectivity kernel, where actually the further you are away, if you're quite far, then there's a cutoff at 60, but you can be connected as well. So you have to make a choice here, a choice for the local connectivity kernel. Okay, so here the surface simulations. Why can this be important? Because as soon as we add a stimulus, often the stimulus in reality is added on the surface. We have a question here. Do you need to create a postulation based on the vertices of the triangulation for the long-range connections? Yes, you do. But often you already do that when you do the normal pre-processing. So thanks for the question. So we have seen that on Micha's slides, going back to the second lecture, where actually when we recreate the brain and all the connectivity, what we're doing in principle with our postulation is already taking every small part of the brain and putting it into regions. So if we want to do that now for the whole surface, we look back into our pre-processing and we look which triangle belongs to which region. And we will do that with the virtual brain also in a few minutes with the hands-on session. Okay, and we have another question. Any recommendations about how to choose the local kernel? Is it related to what kind of phenomenon modeled? Yes and no. So the recommendation that I would give you is always to start with the Gaussian kernel, because law of central numbers, right? As soon as you have enough of those triangles, enough of those connections among the surface, you will be ending up having a near Gaussian distribution of them. So that's the safest choice, let's say that. But for the others, it depends. If you're maybe just interested in a smaller part, you don't have this law. So you have to make a choice. And then, of course, you can try out and see what's fitting best with your empirical data. That's always a safe way. Or you make a choice by really looking into literature and seeing what other people did so far, or even what experiments showed. And there, I would advise you to look into post-mortem data, because they have put the brain in slices and really looked at those local connections a lot. Does that answer your question? Okay, cool. Okay, so I just wanted to add here to this picture that sometimes a stimulus is indeed added on the surface. As we have just seen with the Neuralink, for example, it's adding stimuli quite far above, so on the surface, but also TMS, transcranial magnetic stimulation, is also performed on the surface. So sometimes we need a really good idea, really good model of what's happening on the surface in more detail than just region-based. So that's what this is about, these surface-based stimulus. So let me show you this first study that actually applied the virtual brain and surface-based simulations and stimulation by a former colleague of us. And he gave in, of course, everything that we know: postulation, connectivity, delays, track length, and then also the local connectivity. So here was actually 74 cortical regions and also a lot more, 116 subcortical areas as well. So a lot of subcortex in the study by Spiegler et al. And he added these short range or homogeneous connections and looked at whether this can actually reproduce a stimulus effect in more detail. And yeah, I can already tell you it's like this. So let's look at his results. So what he did, he stimulated one region after the other for all of those regions and looked at the propagation of the stimulus over time. First here, how the amplitude of the stimulus is of course decreasing after time, but then also spatially. If I stimulate the brain here, what is happening to my brain dynamics? Where is it going? Which other regions are also becoming overly active after the stimulus? And by doing this, he actually found that we can recreate all the fMRI resting state networks with the virtual brain. If we just stimulate the right region, then we can actually show that this is lighting up. In this case here, for example, the default mode network is lighting up. And then which regions are those? Of course, you're asking, which region do I have to stimulate in order to see resting state fMRI networks? So these are the classical networks. If you don't know them, that's okay as well. They're quite famous because they're often shown in fMRI. So this default mode network, for example, is lighting up whenever you do nothing. That's our default mode that you can see quite nicely on an fMRI. And there's a visual network here, for example, and all the others. He was able to recreate them all. So in the upper part, always you see what he found from his simulation, and the lower part is how the resting state network is known. So you see quite a good overlap between the virtual brain simulation and real data from fMRI. Okay, so here we can see which areas did he actually have to stimulate in order to light this up. So for the example, for the working memory here, this is cortical area. So there were quite a lot of cortical areas that were able, after we stimulated them, that the working memory network was actually lighting up. And this lower part is all the subcortical areas. When he stimulated those with the virtual brain, then they were also able to reproduce quite a lot of those resting state networks. Only the ventral stream network and the auditory, for example, could not be reproduced by stimulation of the subcortical area. So this just nicely shows us how well we can reproduce reality by stimulation with the virtual brain. Okay, so then another study from this former colleague of us, who's now in Hamburg, just came out last year and was actually about reproducing everything that I've just shown you in mice. So the mouse brain, we can take the Allen Mouse Brain Atlas, which is a very detailed atlas and which includes much more higher resolution data than we have available for humans, because they perform track tracing experiments in mice. And here he did the same thing. He stimulated all the areas and he looked at the response of the brain. He again found that the resting state networks were lighting up very often by stimulating various regions of the mouse brain in this case. So it was a confirmation in mice, actually. And then this wasn't enough. This time he also used optogenetic stimulation data, so empirical data of stimulation in mice, where the stimulation propagation was measured by optogenetics. And this is what's been happening in the real world, and this is what's been happening on the simulation. So as you can see, there's quite a good overlap. This is just one example. You can have a look in the paper and see that there are actually many examples of this kind of overlap between empirical data and then the virtual mouse brain in this case. Okay, so we are, that's great. Okay, I'm through with the slides already, and we can slowly move towards the hands-on part, but I would give some time here for questions on the slides about surface-based simulation, some stimulus. Do you have any questions so far? Okay, I see a lot of people are online. Great. Okay, then I guess we will already, great, slowly move towards our first simulation part. There's a question. Is the activation of the network dependent on the stimulation parameters? Very good question. You will actually be able to find that out yourself during the hands-on part. But yes, in many cases it is. But if you, in my experience, reach a certain threshold, it doesn't matter that much anymore. But it really depends. It depends on your brain, on all the other parameters, on the neural mass model. And this hasn't, I would have to look up if this has been checked in the study as well, that different kinds of stimuli were given. But it's a very active research question that we are actually currently looking into. So I myself, with my colleagues, we are looking into this. Does it matter that all the stimuli parameter? So cannot fully answer your question, but my guess is yes, and we will see in a minute. Okay, let's slowly move. Okay, you're welcome. Towards doing it yourself, because it's very easy. I will show you. And for that, you just need to follow me going, and we will apply a stimulus and make a surface-based simulation. We will need to log into eBrains now, and we are quite a lot of participants. We will try that. Fingers crossed, everything will just work out the way we are hoping to. So please go to wiki.ebrains.eu and then this thing. So can I post this in the chat? Yes, I can. Okay, so you should have the slides and then you can just copy paste it, or you can search for it. We go to the wiki eBrains and then we look into search for virtual cores stimulation. There you hopefully will find the collab. You log in with your eBrain credentials, of course, and then you follow me to copy. So I will do that now very slowly to get you all on board. Okay. We go to wiki.ebrains.eu. You should be logged in. Otherwise, you click here on the upper right side on logging in. You log in. For me, it's automatic, so nothing to see. You can search for the collabs. And you just type in virtual course. And you find the collab space that I created. Virtual course TVB stimulation. And here you can see you can also take this whole link directly, but you should be logged in to see it. So just post in the chat or ask if you have now any questions to follow. If you are able to access this, it is probably because you are on the team of TVB workshops and have been registered on eBrains for this TVB workshop team with Petra. If you're not on this team, you can currently not access, but you can just write an email to Petra right now, ask her to add you to the eBrains group, and then you should be able to access this collab. Okay, now if you can see the collab, that's great. Then hopefully you have access. You can see everything here. And now you can go to Drive. So all the files you can see under Drive. And there you find the notebook for today. Virtual brain _stim. So in case you just open this right here and you do stuff there, it will not be saved and you have no rights to write on this collab. You're just a viewer. So what you need to do is copy this into your library called My Library. So we will do that. We will take it. It's also described in the slides. If you're missing out on any step, you can just look back in the slides. You copy this. You go to other libraries, My Library. So copy this Python notebook to My Library, because when it's copied there, then you can actually write on it and do whatever stuff you like. So then you have copied it. Now you can start the Jupyter Lab. Now we can go just to lab.ebrains.eu, lab.ebrains.eu, and you should be able to see it here. So for me, it's already open. Aha, now I close it twice. Okay, so if you are not sure where to find it here on the lab.ebrains.eu, you go to Drive, your library, My Library, and then you click, it should be there. Great. So can I get some yes in the chat from people who are able to see now this notebook in the lab.ebrains.eu? Can you see it? Do you have it in front of you, just as I have it? First, yes. Okay, great. Yeah, great. Okay, that's cool. So then let's get started. I will show you here for a second, broad overview of what we are going to do. This will be 2 steps because, as I said, surface-based simulations, if you simulate every triangle and then you also want to stimulate that, that's a lot of computational effort that we are doing. So we don't want that. We will split it in 2 parts. We will make sure that you are able to run a surface-based simulation first, resting state, surface-based, and then part B, we will stimulate, but go back to a region-based simulation to have not as many nodes and not so much computational time. So this is split for computational reasons, but feel free at home later on, not now please, to do it on the eBrains with a surface-based simulation and a stimulus. And this should be in the end there, these nice brains. So I've created a lookup table for us where I will post this Google Docs as well in the chat, and you can open it next to it, because this is where we will post our results. And this is for you. Here's our lookup table. So what we want to do is, based on our region-based simulation, we want to put in these kind of screenshots, and everybody is encouraged to do so already now. When you're performing, as soon as you have figured out how this notebook is working, you will be able to select your favorite region. So you could select any one of those. Here are the indices of the region. I've already done region number 4, for example. I've already stimulated region number 12, so the primary motor area. And please just post a screenshot such that we will actually kind of reproduce the Spiegler et al. Study ourselves. And we will have, in a couple of like half an hour, maybe after I've finished explaining and we finished playing around a bit, people will just posting screenshots here. You are able to write on it and edit it. Please don't delete it. And we will have our own lookup table, and we will see the different propagation patterns for different regions being stimulated. And maybe some of the resting state networks will light up, who knows? So for explaining the notebook, I will go through it very slowly. You don't have to post any results yet in the lookup table. You can also just follow me and do it later on. So we are not so many people right now online and on eBrain, so we try to do it together to execute it. First, we have to, import some things. If you do this, maybe on your local computer, this just installs everything that you need on your local computer as well. It installs the newest TVB version. So this whole notebook is also made for TVB 2.1 alpha release. So the newest TVB version that just came out. And you can also locally download it on the last slide of the presentation, actually, all the way in the end. You will find also a local way of installing everything in case you prefer to do it locally. Then we have a surface-based simulation. I explained to you that we will have two parts. So for the first part, just the surface-based simulation with resting state, we will need to load something that you have not loaded before. Probably it's called the Cortex. The Cortex is actually what it is, the whole Cortex. It's actually a 3D thing, your whole Cortex decoded in 3D space, but it will be a 2D representation of it. And this object includes all the information we need to reconstruct on the whole Cortex. So we need to import here a bunch of stuff that we probably don't need everything here. So it's not optimized. And then we need to choose a couple of things for our simulation. We will just choose the generic 2D oscillator, which is non-biologically based model. We often use it just to get some idea of what's going on with the simulation. It's the parameters cannot be placed back or traced back to actual biological phenomena, but it's a fast model to work. So I chose it for our toy simulations here. Then we will load the default connectivity of TVB. If you do this and just fill in nothing, it actually loads automatically the 76 region connectivity of TVB. And then we will choose a speed. How fast do we want our dynamics to exchange each other in the brain? You always need to configure your connectivity in TVB. And we need to choose a global coupling factor, of course. And we need to initialize some integrator. So how is our model actually being integrated? How are our equations global and locally actually solved? So this was very basic TVB, something you have seen before. And now we need, here comes the Cortex later on, and we need a surface as well. So for the surface, we need to really, describe now the local connectivity that we have just learned about. So how well are the regions connected to their actually spatial neighbors? So we will also import some plotting tools, why not? And here is the Cortex. This is the default Cortex. Again, if I fill in nothing in the new alpha release, this automatically loads the Cortex. Default Cortex related to the 76 nodes based on the AAL Atlas. And I have now here a local connectivity that I defined myself, but you can also just do Cortex local connectivity, and fill in nothing, as I just marked here. Again, if you fill in nothing, this will just load the default local connectivity, which is also Gaussian-like. But here we define an actual Gaussian with some sigma, standard deviation, and some amplitude, which we will choose as our local connectivity. And there will be some local coupling as well. And we always, don't forget that with TVB, when you work with TVB, always configure also your Cortex. All right, and then we will need to plot. And this plots it quite nicely, the whole surface in 3D. And you can have a look. I think it looks nice, but yeah, however you prefer. You can play with it, give it some other colors, make it nicer, but this is actually what we're looking at. Now we have all the information to reconstruct the whole surface. Okay. Great, and then we will have a closer look. We had a closer look now at the surface. Now let's have a closer look at what kind of local connectivity I just defined there. You can also choose a default and have a look at the default. You can choose Mexican head, however you like it. So here is how it will look like. So I copy pasted some functions here, which I couldn't find in the new alpha release. That's why it looks a bit messy. But what this does is basically just plotting to you how your local connectivity looks. And this is nicely Gaussian. So this is how, again, each triangle of the surface is connected to its spatially in millimeter close neighbors on the surface. So for TVB, we also need monitors, because if you're running a simulation and we don't have a monitor, nobody is monitoring. So out of your simulation, nothing will come out. Great, you ran a simulation, but without anybody writing down anything. So you always need to specify in TVB the monitors. So this can be, now my explanation can be very repetitive. If you're already a total expert on TVB, you can zoom out for 5 minutes and fill in our region stimulation lookup table, right? So here, for the monitors, we will take a temporal average monitor, a spatial average monitor, and here is what was asked before: how do those triangles know to which region they belong to? It's via this region mapping. And again, I filled in nothing. So for the new alpha release, that means it's loading the default region mapping. So this region mapping is describing for every triangle to which region does it belong to, so that later on we can nicely find this back for a region as well. And we will actually monitor some EEG out of the surface-based simulation, which is quite handy, and it's natural, right? EEG is on something measured on the surface. So if we give it a lot of information on the surface and make it very realistic on the surface, we are hoping to get very realistic EEG data. So we will take those 3 monitors together in a bundle, and now we are going to simulate. If we do this all together, I don't know how long this will take, but let's try it out. Now we have our model, which was an oscillator, which was just a generic 2D oscillator. White matter is just a default connectivity. We have some global coupling. We have an integrator function of how to solve those differential equations. We have the monitors, and we will have a short simulation length. And here, this is what you have probably not done before. We load the Cortex CTX as a surface. And then when you click sim.configure, this is running your simulation. Oh, it went very fast, so we are fine. You can all execute it. Okay, so now we ran the simulation. Here we need to write everything into the simulation and actually get out our monitor data. Ah, yeah. Sorry, my mistake. So this is configuring the simulation, and this is running the simulation. Of course, it takes a bit longer. So in TVB, what you have to remember, always configure first. Okay, so as soon as this is finished, we can print some pretty pictures and look at this. So this is the regional average. So we have one line for every region, and we look at how this is the activity here, generic 2D activity. So just some oscillating activity is going over time. You see, we have a much too short simulation here to have anything realistic. Same for EEG, but of course, for computational reasons, I made it very short. And we have here some data that will now, yeah, hopefully, if you perform much longer and with a more realistic model, you can get some very realistic looking EEG data here. So if you have follow this like I just executed, you have performed your first surface-based simulation. So to sum up, what was important for surface-based simulation, especially, so actually it's only this one line. You have to load a Cortex. Okay, you keep getting an insufficient memory error. We're trying to start my server in lab. Okay, it could be that it's limited to. Oh, I'm so sorry for you guys. So you cannot even see the notebook, but then if you can see the slides and you go to the last slide, you can download the notebook onto your local computer, open it there, and then just don't comment those lines, but leave them, which should install everything, the newest alpha release. And then you should be able to just do it locally. There's a whole explanation on local. It's in the slides all the way to the end. How to run everything from today on your local computer. So this is what I would advise you to do. Yeah, just try it out. Let me know how this works. It can be that we are too many people. Then I'm really sorry. This is what you're working on a big server, but I will leave this open as well, the region simulation. Hopefully we will get to the nice lookup table soon. And after I explain how a simulation is performed, and then you can do it also after the session. Okay, so the surface-based simulation part is with that actually over. Great. And we will not put a stimulus on the surface. Too expensive for the moment. People are already getting memory problems. That's not a good sign. So, but we will still try now to perform your first stimulus, simulation with TVB. So here we will just, what we will do is just load the default connectivity from TVB again. And now we need to define a stimulus. So we need to define a weighting. The weighting is just a vector defining which region, in this case, I chose region 4, for example, which region is actually given a stimulus. So for this region 4 is given a 0.1 stimulus. So this weighting is a bit arbitrary. You have to play around with it, but you can also, it's made because you can then also give for different regions different weightings of the same stimulus. So for which case can that be important? For example, Transcranial magnetic stimulation, TMS. Because when you hold a TMS device, creating a magnetic field onto your skull, you will not only reach one region. You will have a spatial distribution of reaching with different weightings, different regions. All the neighbors will be also stimulated, less, but they will be. So it's hard to target one area with this device. So for now, we will just keep it and stay with one region that will be stimulated. Actually, I want region 12. It's a motor cortex. I like it much more. Okay, so the temporal profile of the stimulus, we need to give it more information. What kind of stimulus should be identified? So for that, we would choose a standard pulse train. It's just a standard going up, giving a pulse, going down. It will have a certain onset, in this case at 1,500 milliseconds. And T and tau are the two parameters that you need to be aware of here. The pulse train will have a length of 50 milliseconds. So for 50 milliseconds, we will give a constant stimulus here. So tau is a parameter for the length of the stimulus. And then T is a parameter for every repetition time. So actually this stimulus here defined here will come back every 1,000 milliseconds. So it will start at the 1,500, give for 50 milliseconds a stimulus, go away, and again will come back after another 1,000. So we need to give it this information and define the stimulus, and then we put everything together with this function patterns dot stimuli region. And then the equation, the connectivity, the weighting, everything has to come together. And we can have a look at the stimulus and look when it's configured in, always configure in space and time. So as you can see here in time, it will look, if we have only a short simulation, it will look something like this. Okay, there will be a stimulus on one region. And in this case, the space, these are all the regions. This is region number 12. A stimulus weighting of 0.1 will be given. Yes, this was exactly what I have given as an input. And from the time, this is milliseconds, it will start after 1,500, give for 50 milliseconds the stimulus, go down again. So yes, this is what we are making. And here's time and space together. It looks a bit weird, but because we just gave now one small little stimulus. Okay, so you can have a look. This is the stimulus that we wanted. And now let's perform your first stimulus in the virtual brain. Ta-da, it's happening. Again, I chose as a model the generic 2D oscillator. Now with some non-default parameters, I chose the default connectivity, global coupling, an integrator, and I just chose the temporal average here as a monitor. Okay, so we performed it. Now let's have a look at it. Okay, this is how it looks. So in red, you can see, so K is red. You can see here the region, the average of all the regions. And you can see in black, plotted on top of each other, all the regional reactions. So what can you see here? We have a long simulation, right? And here, whenever I said 1,500, give it a stimulus. Yes, it gives a stimulus. And you can see reactions, so these black things. And then again, after 1,000, give it again a stimulus, so that's at 2,000. Okay, we can see that. Yeah? I hope you can see that as well. And for the average, it also changes a little bit. Like we would have to zoom in very much to have a look. But it's much more beautiful to visualize now that stimulus propagation on the brain and the surface of the brain. For that, we will load some helper functions first. So here the credits go yes, great. So here we can load this helper function, and then we will give it all kinds of things, and also the cortex, so that it knows which region will be plotted on the cortex where, and so on. Trust me that this is correct, but here, for example, we give it also a minimum and a maximum of this activation where we'll cut off, because maybe you don't want 0.01 things plotted on the brain. Maybe they're too small and you say, oh, that's not in reaction to the stimulus. I only want to see really the stimulus reaction. So here the time steps, we know at 1,500, we are giving the stimulus. So what we want is just after the stimulus, look how it's propagating. So it lasts for 50 milliseconds, so it will still be here when we are looking at time point 1,505, and then we will see its propagation. So let's see that this will work. And I remind you, what we did is we just stimulated the primary motor cortex here in my notebook, which was region number 12. Yes, I can see the motor cortex lighting up. What we did was stimulate the right motor cortex on the right hemisphere. So we can see it here nicely, the right hemisphere. And we can see it's there and it's spreading. You can see regions lighting up as far as the temporal cortex goes. And more interesting, even when we look from on top of the brain here, that's the top view, you can see that directly the left motor areas are also lighting up. You see that even strong? Yeah, that's interesting. So they are closely connected, right and left motor area, right? The coordination between your movements between right and left and so on. So it makes sense. And this is how it propagates. And this is what I already put in the region simulation Google Docs. So here, now for everybody who can perform, who got a server running, I suggest that you stimulate a different area. And we will do that. I will do that together with you as well so that you can follow me. I encourage you to choose your favorite area. Let's see. Where do we have to go? That was surface here. Okay, here we had all the 76 areas, and here I chose the motor cortex. So now you check and you see, oh, maybe I like, what do I like? Amygdala, the center for fear. Great, let's try this one. It has the index 2, so let's stimulate that one. And you don't choose 2 now, choose another one, so we will get pretty pictures. So the only thing you have to change is here in this cell, spatial pattern of the stimulus, you put in your favorite number or region. I put in 2 now. And then you go run, and let's run all cells. And then come back to it in a second and make another screenshot and fill in our stimulation table. So for you again, try out if you can perform a stimulation of another area. Open the Google Docs and put a screenshot for us there. Choose any area, doesn't matter. And run your simulation. And then, of course, we have to wait a sec. As this is running, are there any questions for how to perform a stimulus simulation with TBB concerning the things that I just showed? Feel free to ask. Okay. So for me, the whole notebook just ran. And I got another pattern. I now stimulated the amygdala, center for fear. And I got, with the same pulse train, same stimulus, everything same. And I got this here. So I'm making screenshot and plugging it in here. And you can plug it in as well, whatever you just stimulated. It looks very similar to this one. Okay, and then we can create together a lookup table where you can see how the stimulus would propagate. Great. I'm seeing that the RV1 has been done. Great. Can you post a picture for us in the just to the right in the Google Docs? Ah, here I see it. Yes, thanks. Okay, great. So we have another area stimulated here. Same pulse train, everything same. And it was running, it's going well, so that's good. Good to know for me. Great. I'm awaiting more areas here. So you yourself can see whether you are whether you would have expected this or not. Of course, we cannot see subcortical areas on this small surface here, but we can think about whether this is a realistic, hey, great, the parahippocampal cortex here, area 24 has been done as well. Great. And you see some of those, hey, super, great. So it's working for people. That's great. So when you compare them now, looking from further away, for example, these two, like the area 18 versus 24, you see some similarities. Some patterns are lighting up whatever you do and stimulate. And this is probably because, so I'm a network scientist, these are hubs, right? If we look here, motor cortex, it's a huge hub in the brain. It has many connections. So whatever other area you would start and stimulate, this stimulus is probably propagated through the motor cortex as well. That's why it's lighting up as well. But you can see other parts as well lighting up, and we can create together a cool lookup table here. Yay, it's coming together. Okay, awesome. So that means your simulations are running, you are able to simulate and actually have your own beautiful brains there. Cool, so you can do this at home as well, fill it up later. If it's not working for you right now, don't worry, there are enough areas for people. But it's a cool sign that it's working for everybody and that people are able to perform it. So you have learned to perform a simulation simulation, how we call it, in TVB. Great, I'm happy. I hope you are too. And now let me just show you some additional optional. You can change the parameters of the stimulus, right? So let me do once here and choose, as long as people are maybe still running. I would just choose a different local connectivity and show you how the default local connectivity that I will just uncomment here is actually, we can have a look how that one looks. This is the default local connectivity, just in case you were wondering how it looks. So it's a bit more narrow. So we broadened it a bit to have a more, yeah, widespread surface simulation here. And this is the default connectivity. And you can also run a simulation here with the default connectivity and look at the results. Oh, okay, good to know. Yes, that can be. So let me share it again with you. I included it slowly, and then you have so many versions and so on. So try if you have it here, because if I share it now and I have it here, it should be in there. So people are looking for a slide for the local, how to install everything locally and just run it locally in case you want that. Here it's included. Okay, so please try that link. Only the last 2 or 3 slides are different, but they're important if you want to run it locally. Okay, great. How is our lookup table coming? Awesome. Great. And basically what we have been doing here is reproducing the Spiegler et al. Study, which now we could try to identify, average maybe over the whole time here, that the stimulus propagates, perform it multiple times, and then compare it with our resting state networks and see if we have a resilience, a resemblance with some of those resting state networks. Okay, great. So local connectivity, default connectivity, I've shown you can adjust the settings of the stimulus. Of course, you can adjust them. I've shown you where we do it here, for example. If you want the stimulus to start much sooner, if you want it to be much more often, okay, that's too much maybe. If you want it to be much more often, you fill in and change the T, for example. So you can do play around with this, do different things with it. You can also load, what I included here is not the default connectivity with 76 nodes. Yeah, this is 76 nodes, but you can actually also load exactly the one that the Spiegler et al. Study used, which was one including also many more of the subcortical areas and having in total 192 regions. So feel free to also reproduce with the paper exactly this whole lookup table that was created by a previous study from Spiegler et al. Okay, so I guess we are through this a bit. People have figured this out, great. first learning goal done. Now, there is another thing that I wanted to show you, which I haven't shown before. So we now know how to give a stimulus to one region. We now know how to give the same stimulus to multiple regions. But what we don't know is how to give multiple regions different stimuli. But this can, of course, happen. If, for example, you create some magnetic field, some electric current field, however you want to place it, and it alters with distance, or you have an electron, there are two different contexts, and they're giving different time courses. So in that case, what you want to do is you want to load this class, multi-stimuli region class, where you can give multiple stimuli for different regions with different time courses. And then in that case, this is just the definition of the class. You need the neo trace, you need the new alpha version, great, it's a new feature. And then this is loading again the connectivity, default connectivity. And then we will make this pulse train, and we will have 3 different trains here. And we will put them on different regions. And we will let them begin and stop at different times. Yes, we have here, but because they have different lengths, there will be different timing. And then when we look at them in time and space, as we had it before, we can see a beautiful picture here, where we have once a stimulus that is quite long, same onset time, and a stimuli, 2 stimuli here that are shorter. One is even more intense than the other one. Is there a convenient way to look up which region has which label or number in the notebook itself? Yes, good question. So that's why I put here the region number to make it much more easier for you. So these region numbers, I started at 0, like Python, so that you will have the right number. If you type in 0, it actually will be stimulating the primary auditory cortex. Does that answer your question? And these are from the notebook itself. This is already the index that you would have to fill in. So it's very easy. Inside the code without an extra table. Oh yeah, that's a good question. So once we're inside the code, is there a way to look up which region has which number? Yes, if you look into the file centers, so you can go and load here the default connectivity, and then you can have a look at the con dot centers. Now I always, yeah. Okay, centers are just the centers, but the text file which loads the centers, the region, it must be region names, has no, so it's something like this. So whenever you are loading this, you can also just check out the TPP database, and you can go and find the file centers, because in the file centers, the first column has the abbreviations, which would for every region, which is then this column. And it's probably loaded somewhere as well. But sorry, I can't give you the right command here. But if any of my colleagues who are online know the right command or want to look it up, feel free to do so. Because then you get a whole list of region names. But depending on what you loaded, maybe you only get abbreviations. And then you would still need a table to look up what the abbreviations mean. But I made you a table for this, and this is for the default connectivity. So you are safe as long as you're using the default connectivity of TPP. Okay, so here we can see how we can perform then. Now we can give this stimulus here that we just defined, just in our normal simulation. And then we would have a simulation which performs different stimuli with different strengths, different timings, and so on different regions. And where do you put the stimulus? So just to summarize how you perform a stimulus, regional simulation, never forget this line. Define your stimulus inside your simulator. So basically, if you are familiar with TVB already, everything you need to know for surface-based simulations is define your surface in your simulator. And for a stimulus simulation, define your stimulus inside your simulator. And then you're good to go. And you have learned 2 more skills. Okay, so we have seen this, all great. We have tested a bit how it could look like. We have seen it on the brain. How's our lookup table doing? Great. Okay, I don't see any new postings. Okay, so I think people who wanted to do it, who were able to get an eBrains thing, they were able to do it. So that's great. Now, this is the homework. I will come to that later, but feel free to already have a look and feel free to adjust everything that you want to adjust. So if we play here with the stimuli and you maybe want to make it very realistic, right? I told you about TMS, for example. You would just have to figure out what's the spatial distribution distance of a realistic TMS device. Where is it often placed? So prefrontal cortex, for example. And then you can already perform this with the virtual brain and look how, in this case, the default brain is reacting to TMS and try to get as close to, if you have data, as close to data as possible. And you're able to do that all without harming anybody, right? That's great. You can also load the mouse brain here, which is track tracing data, so very reliable data. And you can stimulate the mouse brain of the Allen Brain Atlas. There's also a mouse brain as one default connectivity in TPB, which I think is quite interesting as well. Okay. Are there any questions on the notebook excluding homework? Anything was too fast here? I hope you were able to follow. Hope you all can see those nice patterns. Okay, that's great. So then let's come back and see I would just remind you once again that we have a Kahoot quiz on the site. And this is a perfect timing for you to start and answer the first couple of questions because we are through with the first part of the lecture. 5 minutes early, so it's time for questioning. Or try and spend some time on your Kahoot quiz. It's open until 6. Just reposting so that you don't have to do it. The slides you're hopefully able to find. The notebook running. So then I will maybe just go for a minute here through the instructions if you want to do it locally. What you would need in order to run the new notebook is namely the new alpha release of TVB. So you need to go to this website for finding the new alpha release. And maybe everybody wants to download it, who knows. It's great to have, I think. For your specific operating system, you download it, and which this does is basically downloading a folder called TVB distribution. This folder will probably pop up somewhere in your downloads, and then in your downloads you will find a DMG file in there, at least for me, for Mac. You double click on it, this installs the TVB app actually, and you just have to move the TVB app to the applications folder for Mac users. And then you will have the new alpha release. And if you then want to start the Python interface locally, so on eBrains, we have seen it's quite easy to start, right? And locally is also easy. You just go to the subfolder bin of this TVB distribution, so this downloaded folder, and there's something where you can click on called Jupyter_notebook.command, which then opens and starts your local host for your Jupyter notebooks. And then you will find a folder in there called demo scripts, and this will actually automatically open the folder demo script. So you will find also a lot of demo scripts in there. So I have made you this demo script here, right, on surface-based simulation and stimulation, but there are many more. And I'm, for example, interacting with Alan, which is one for the Alan Mouse Brain Atlas. And you can, everything is well described. There are all Jupyter notebooks to explain to you the code of TVB and how you can actually put it to use. And then just to finish this, you can just move the specific notebook. So in this case, you need to download the course notebook. I will just post this. If you download this course notebook to your local computer, put it in this folder demo scripts, and then you will be able to run everything. Yes. Okay, so there's a question related to the surface again. My cortical surface has 16,384 vertices, right? That are all the little triangles. But the default global connectivity has 76 vertices. Yes, correct. Each region here is a vertex, or if you go surface, each triangle is a vertex. Before you said that you would need to compute the long-range connectivity for each vertex. This doesn't add up. Can you explain what's going on? We need to compute the long-range connectivity for each vertex as well, because, okay, I think I see where you're getting. Okay, so region-based simulation, you have 76 regions. You need the regional connectivities, which we call long-range connectivities, right? And now this just adds to it a surface-based simulation. So now you have all those 16,384 vertices, but each vertex still knows, due to the region mapping, to which region he belongs. So this small vertex will be locally connected with the local connectivity, but will still also be connected long range with other vertices belonging to the other regions where he was connected before. So it's not that this long range connectivity that one region had with the other one is now gone. It's just added. So you have 2 times of connectivity. I know this can be very confusing. So you have the long range connectivity stays the same. Before it was one region connecting to the other region. Now it's all those little triangles of this region will connect to all the little triangles of the other region. That's what I meant when I said we still have to compute the long-range connectivity for each vertex. Does that explain it a bit? Otherwise, just feel free to get into discussion here.
Yeah, hey, can you hear me?
Hey, yes,
Yeah.
I can.
Thanks for explaining. So yes, that answers kind of what I was getting to before. But
so it's still there, the long-range connectivity. Yeah, it's still there. Nothing changes with the long-range connectivity.
Right. So okay, cool. Because what I understood before is that for each vertex of the cortical surface, you would need to estimate probably from the DTI specific weight, because that's why I checked it again, because it wouldn't make much sense, because then the computations would be humongous.
Exactly. If you want to aim for that.
Yeah, and but so you can use a downsampled long-range connectivity, as in this case with 76 vertices. Is there a way to also introduce some sort of long-range kernel then?
Yeah, of course.
Right,
You
because
can do
because
your
I guess
own,
it's
yeah.
some sort of unrealistic that this a huge region out of 76 regions, they would get a homogeneous distribution of connection weights.
Yes.
Does
So
that make sense?
I'm not sure I understood your last question, but
Yeah.
maybe can you do it again? Yeah.
Yeah. So what I mean is, I think the way you explained it right now is that let's say we have one region out of the 76, and
Yeah.
that contains some 1,000 surface vertices.
Yes.
And they would have all the same weight to the other region. Right?
Yes, to the other region, right? Yes.
Exactly. But is there a possibility to introduce a kernel such that, for example, the surface vertices in the periphery of that region get weaker inputs, while the center.
Yes, of course.
Uh-huh.
Yeah, so yes, it's possible. Not in the form that we are loading it now. That's not possible. There's just assuming, but you can. That's the beauty of TVB, right? You can adjust it yourself because it also is more realistic. What you just said, right? There's not just one white matter connectivity going from everybody exactly in the same way to everybody else, right?
Right. Do you happen to know what the commands would be for that, or what would I have to look into in the documentation?
So one way to do it, how we are currently doing this, is using the TVB multi-scale. So we want to zoom into a region, and in that region, we want to really give every neuron connectivity to every other neuron. And there you can specify it really per neuron, what connectivity you want it to have. That's one way.
Right, but
okay. And that's a second course then. But if you want to have it just spatially and have it the same for every region, I will have to look it up. And but of course, I will look it up and let you know.
Yeah.
Maybe it's cool to write an email as well, that I have your email then as well.
Yeah. Okay. Cool. I'll do that.
Write it to Petra. Write it to Petra.
Okay. Yeah. Thank you.
Yeah, you're welcome. Cool questions. Okay. Let us take a few minutes of break. I suggest 10 minutes until 43. And feel free to try out and run this now in the break, however you like it. Fill up our lookup table. Oh, cool. Even more things coming up. And feel free to go on with the Kahoot quiz, or just like me, take a real break, and see you in 10 minutes. Okay. Great. Now we're back. I hope you had a great break. Got something to refresh yourself and are ready for the next, not that much anymore, little less than 1.5 hours, where we will dive into more cool stuff about brain stimulation. So I saw 7 people loaded up their propagation pattern already, so that's great. Seeing that it worked for so many people simultaneously, it's quite a success. And now first, let me show you what's happening now for the next hour and a half. Okay, so we are back for the next lecture part, which I will cut off at around 5:30. And then we will go to another hands-on part where we'll first probably explain the homework a little bit. And then we will have a look at the graphical user interface so that you can even see a very cool video, hopefully, on eBrain of your stimulus propagating through the whole brain. So for that, we will dive into my favorite form of stimulation, which is deep brain stimulation, or shortened DBS. What is deep brain stimulation? Not last time, but the time before, you have heard already about this kind of stimulation, where I presented to you a use case of the TVB multiscale part, where we were looking in the basal ganglia in very much detail, and in the nest simulator, and we actually, as a homework, you were performing already virtual deep brain stimulation, which is also my main research topic for the moment. So just as a reminder, what is deep brain stimulation? So we're talking about this small device here, 2 electrodes being planted into a person's brain with it's invasive, very much invasive, during a surgery where, let me place you here, where there are these electrodes implanted and configured during the surgery. And then there is a cable going all the way to the chest, where there's a small pacemaker, kind of a pacemaker-like device, which has the batteries and so on. And this is actually giving, there is also a remote control as well, which can program this small stimulator device, and then it's giving up some electrical currents and pulses around its location. So where is my mouse? Here. So for here you can see one of those DBS electrodes, which is also called the lead DBS, implanted into the STN, the subthalamic nucleus. And then here it will be activating this region and around surrounding regions. It's a very famous target in Parkinson's disease, the STN, the subthalamic nucleus, but also the GPI has been targeted successfully before, but it's becoming less and less popular to use the GPI, but there are still centers doing so. And there's ongoing research into which area is better relieving Parkinson patients from their symptoms. So what are the advantages we, as I just said, have, for example, in Parkinsonian patients? We see that their tremor, for example, is becoming less and less when we insert an electrode in this way and insert it in the correct placement with the right configuration. We have a successful case of DBS. But as you can see here in the picture, there are also some challenges coming with deep brain stimulation. You're targeting structures which are very deep inside the subcortex of the brain, very deep inside the brain. So for the surgeon, the placement also is not always that easy. It's totally possible. They do reconstruction and simultaneously on a screen as well, so they see perfectly where they're heading. But often, as you can see here, for different patients, if we reconstruct afterwards, the placement has been different, and which is normal. We are all individuals, right? But for some patients of those, it worked out well, and for others, it didn't. So it seems that the placement, and that's a very common research result in this area, is a very important factor, whether your stimulation will actually be helpful for the patient or even be harmful. So it is a treatment option, as I said already, for Parkinson's disease. It's approved, it's being done here in Berlin at the Charité. There are patients coming in all the time, getting implants, coming for a second optimization of their parameters. And it's also been used for dystonia and essential tremor. So for these kind of diseases, motor movement disorders, we can see benefit by implanting DBS. And it was also investigated in the context of many other diseases. For example, as I would have not thought before I came into this field, obsessive compulsive disorder. So for OCD, people, as soon as they get an implant, so after a few weeks of the implant, sending out the stimuli continuously, they already have not so much problem anymore, and they are getting better and better. So this is great. Same has been shown for depression as well, and Tourette syndrome, Huntington's disease, so addictions, cocaine addiction, for example, different other, for opium, it's just been out a study, as I've seen, Alzheimer's disease as well. Chronic pain has been targeted because, yeah, it makes sense that those people are just relieved to have something that's somehow circumventing the normal structure of the brain, which would be sending a pain impulse. Okay, so here you can see, as I said, for Parkinson's disease, the DBS often targets the STN, the subthalamic nucleus. And for red fibers here, you see that this was actually good when they were hit in this reconstruction. And blue fibers, it got worse. And for the same was for, so it really depends the placement. As you can see, the fibers are really close together, and one is giving you clinical improvement when you hit it with the electrode, and one is not giving you clinical improvement and can be even lead to worsening. So this is a problem and a challenge. And at the same time, you have here for OCD patients, as I just mentioned, you have the target area of ALEC, and you can see that also there the fibers are intertwined a lot. So it's a real challenge which fibers should be hit and which should be not. And there's always some surrounding areas which are activated as well. So we have to be really careful with the placement, and it's a challenge, right? But there's a toolbox called LeadDBS developed by Andreas Horn and our colleagues here at the Charité. And they are actually reconstructing after the surgery with their toolbox very precisely which fibers have been hit and where was the location of this electrode actually. And then they can relate it to clinical improvement and clinical worsening of patients in order to give better advice for the next surgery. So as we have seen the challenges in deep brain stimulation, we have very small targets, these structures deep inside the brain of the basal ganglia, where we have a very complex anatomical architecture and the need for absolute precision, because as you've seen, few millimeters, not even to the left or right, can make a huge difference to the dynamics of the patient's brain. And on the plus side, of course, those electrodes being implanted give us very nice data as well when they can read the data as well from the surrounding structures. Because normally when we have an MRI, right, you have data being extracted somewhere from here, MEG, EEG, and you don't get such high quality data of the structures deep inside the brain. Yes, and we have seen it's an effective treatment for many people. And it can help a lot and really change lives. So I'm quite fascinated with it. So I hope you're too. So it can also do other things. We have talked about relieving symptoms and in this case, motor symptoms of Parkinson's patients. But recently, our colleagues here at Charité also showed that it can change your behavior in risk-taking, whether your electrode is on or off. So here they have made a study, Ehrman et al., where they had a game people were playing this game, and the longer you stayed in the game, the more risk you were taking, but also the more you could earn, right? So that's always a trade-off, when to stop gambling. And the Parkinson's disease patients, whether their DBS was on or off, are here less than the controls, and this certainty equivalent is actually a measure of risk-taking. Yeah? So they showed that Parkinson patients were actually more risk-averse, so they didn't want to take so much risk as the healthy controls. But once their electrode was switched on, this is a measure for being willing to take a risk, was normalizing. So by turning on the electrode, their behavior in the same game changed. Here, this is shown for one patient being once electrode on, once off, and you can see his behavior in the game. Multiple different games were executed. Overall, we're really following different trends. So Parkinson patients are more risk averse here in general, but once their electrode is switched on, they seem to normalize. And this can be partly due to, because for this patient, for example, the VTA would, which is a volume of tissue activated or by the DBS electrode, actually was not only hitting motor areas of the STN, but also non-motor. So that could explain that those areas that were not directly only connected to motor, but also other areas of the cortex, that could explain that this placement of the electrode made the whole difference whether this patient was actually changing in behavior or not. So quite a cool study, I think, looking at other effects that can be changing with an electrode turned on and off. So we know that it's good for movement, but maybe we don't know all the consequences yet. Okay, so the goal for my personal research project, and also what you have done in the hands-on session right now, is to virtually perform stimulation. And in my case, virtually perform deep brain stimulation. We can see also here another picture for the STN. Multiple different placements will be reconstructed by lead DBS, so we know exactly where the electrode is. And we can even use, and you have heard me talk about this in the second session, use TVB multiscale to get an even closer look into where the exact placement of this electrode is, which neurons are actually really activated by an impulse sent from this electrode, or which are not. And of course, the STN or GPI debate and everything, this could help, the virtual brain could really help here to explore different pathways leading to the motor cortex, to really explore, let's say in slow motion, where is our stimulus propagating, if we send it from here or from there, for an individual data, for really an individual with its MRI data, DTI data, Fmri data, maybe even LFP data, the local field potential data, maybe even recorded by the electrode. We have lots of data available, and we can tailor this to make a virtual brain for every person, right? That's what this whole course is about. And then we can tailor for the individual, where should we place the electrode? How should we configure this electrode? When should it be on? When should it be off? And how should it look at all? And normally this is done during a surgery, and this could be even done virtually without a surgery or anything. And then we could already beforehand maybe say, for this individual patient with this brain structure and this Fmri resting state networks, we would be not advising to do DBS, but rather TMS, repetitively transcranial magnetic stimulation, and so on. We could try out different techniques by combining those two softwares. Okay, so here you can see that is a video I've shown before this, just to show you how a stimulus propagation with the virtual brain can look like. We can directly make such a video, and we will actually do it. We will have time for it, luckily, today, to show this to you. And then you will have this video on your screen yourself. But this is focusing on only the subcortical structures. And there's a lot going on subcortically already, as you can see, when this is a simulation of stimulating SDN in a specific patient model. So this is something you maybe remember. This is then one small little extra part about my personal passion here in this project. That's we have TVB for the whole cortical area. We have here the connectome, we put mean field models on top of it, you know all about it now. And then we interface it with a much more detailed model for the basal ganglia, because inside the basal ganglia, we have everything happening, for example, for Parkinson's disease. We know what the thalamus is doing. We know that the STN should be probably stimulated, and maybe even the GPI. And we know from literature and also from rodent experiments how the connections look like, whether they're excitatory, whether they're inhibitory, and so on. So why not use all this information gathered to make our network and our virtual patient even more realistic, right? That's what this is all about. So we build this with Nest or Anarchy. So these are simulators that you have seen before. Nest, at least, Anarchy is similar, where you can, in much more detail, go and give maybe even every neuron specific behavior and then stimulate it. And you can zoom in for this. And then still you can see the effects on the cortex, especially, for example, where you would be interested, the motor cortex. Or maybe if it's about risk-taking, decision-making, maybe the prefrontal cortex would be interesting as well. Why is it changing behavior if we perform a deep brain stimulation here on the STN or the GPI? So we hope that we can answer those questions by building a very realistic model of the virtual patient. And here you can see results already from a simulation. There's no Nest now involved. There's no Anarchy involved. This is just the virtual brain, but very good underlying anatomical data that includes already all the basal ganglia structures. And on the left, I performed an STN stimulation, and you can see how it propagates. And on the right, I performed a GPI stimulation. And I have looked at this for a long time, and you can as well. There are slight differences. Now you see it a bit one after the other. And they're there, and you can make a study now and just checking out for each individual patient which one is better, a better target here maybe. And we can try it out beforehand with the virtual brain. Here I just used the generic 2D oscillator. So it's not very realistic yet and so on, but just to show you how easily you can have a look in real research-related questions that are interesting right now in the field by just performing TVB simulations with the tools that you have been given. Okay, so this is just to summarize and what you can do with it. We can apply a virtual DBS stimulus. We can look at this propagation pattern as you have done and helped me with the lookup table as well. And then we have the different propagation patterns for different region stimuli, which we could also link back to different patients as well and individualize them. So saying that, for example, for this patient, we have this propagation pattern, and we know he has performed DBS surgery, he has undergone DBS surgery, and he was getting better. So we can link his clinical improvement, hopefully, to our parameters used in TVB, optimized for his individual data. So linking always clinical outcome with something that was predicted by the virtual brain, this would be our ultimate goal, right? Okay, I hope you get the idea of this, and I hope that very soon I can present you the real results. Is it really realistic to decide for single subjects whether to stimulate the GPI or the SDN based on the TVB? Has this been verified? Good question. Thanks for that. So, is it realistic? Not yet. It's the ultimate goal, but it seems possible by the tools given. You can do it when you have lots of data of an individual patient. You can tailor his dynamics of the virtual brain very close to his dynamics going on in the actual brain, right? And then, at least from the data that we can gather, we can say that. And then we can have the virtual patient in the virtual world, right? And we can do with him try out the tools that we're given. So we can stimulate SDN and GPI. And we can, of course, not do that, let's say, for long-term effects. We cannot let the simulation run for a couple of weeks or so, like he would be given a couple of weeks of stimuli before people would maybe decide that this is really not a good fit for this person. But we could get very close to this, and we would have to check, and this has not been verified. This is my current research project. But feel free to verify that for me. No, kidding. Feel free to work with us together, and if you're interested, always write Petra an email. I'm happy to take you on board for this project, because it's still very exciting. It's part of a very big project of the German Research Foundation that was just kicked off, the Retune project. It's for the next 4 years, people from Berlin and Würzburg. And I think you're on that as well, right? So we will keep in touch on that, and hopefully I will present very soon the real results on this, because verification is a crucial step. I totally agree. We have to verify this for now with the data that we have at hand. So what we can do is just, as they doing currently with epilepsy, they're just advising the surgeon for the epilepsy surgery beforehand. So for the lesion, which areas do they think from the virtual brain side can be interesting? And already that can be quite helpful to have just a second opinion, simulation-based opinion. Also for the surgeon. And then there's a trial right now ongoing with epilepsy and the virtual brain, and we will see how helpful it is. Yes, great. Yes, so I'm looking forward. I've seen that already. So we will be working closer together already. Then feel free to reach out also via Petra, and we can discuss for many more hours on this. I hope it's realistic, and I hope this will be verified. Okay, so just to show you something for a motivation of then explaining to you the homework later on as well. Another recent study by our colleagues here, with whom we work closely together, showed that DBS was leading to a normalization of functional connectivity. So the functional connectivity of the patient got closer to the control, the age-matched healthy control. So they showed this, and this is actually quite cool to see that this means for us that the whole dynamics of the brain as a system move from sick to healthy by using deep brain stimulation, which is quite cool, and which should be something that we should be able to reproduce. And people have done so. I will show you in a second. And in this case, normalization meant that the patients needed a higher average functional connectivity in the motor areas. So it was somehow too low, and they needed a higher one. And this can be motivation for the homework. Okay, so the newest trends here in the field of DBS. Very excited to show you that. Need to check the time from time to time. Not getting lost here. Is adaptive DBS. So I don't know if you've all heard of it, but it's a very cool approach, because what is adaptive DBS all about? It's not only deep brain stimulation, but it's, let's say, the energy saving version of deep brain stimulation. You only stimulate when it's needed. So for example, just before you have the intention to move, you would stimulate. People are working on that right now at the Charité. I've just seen cool first results on that you can measure by reading the data from these electrodes. You can measure when a person has just the intention to move. And then you would give a small stimulus, which then helps the patient to move. Because without it, he would maybe have bradykinesia, so slowness of movement. But with it, he would be moving very normally. You can watch a lot of videos on this, how the movement changes when DBS is turned on, for example, for, in this case, Parkinson patients. So how this closed loop system of adaptive DBS is working is that we have the electrode placed there, and it's activating some cells, and we can measure these local field potentials just from the electrodes. And then we have some control variable. So we will read out some variable of this data. And we will then analyze it best in real time, right? So no delay here. You want real-time feedback. And you want to calculate new parameters and then give a stimulus. And this is closed loop because you can actually directly alter brain activity, right? You read the activity, you give a stimuli, you change it, you can read back whether it is actually doing what you were intending it to do. So this is a very cool closed loop system, if you look at it from a systems theory side, and where you can test out many different strategies as well, right? And there is very exciting from Medtronic, which they developed such a device. And Medtronic just launched its first adaptive DBS trial on Parkinson's disease patients. So we are very excited. So they will implant this device, which will be adaptive and only be turned on when they think it's needed. Based on many different parameters, they will try out different strategies. And they already started now. They just got approval that they can do this trial, and because once it seems to be as safe as DBS, normal DBS, and second, it seems to be also saving more of the battery life and the energy, and for the patient also better, because sometimes stimuli can be harmful, right? So you maybe don't want it all the time, only when you need it, yeah? So that gives also some control back. And what's the control variable here that they're actually looking into? And this is the beta band activity of the STN in this case, when you have STN DBS, because I found that there's a correlation between the STN beta activity with bradykinesia and rigidity symptoms. So it seemed that there was a higher beta activity when you had the symptoms, and the treatment, so this DBS stimulus, actually reduced the beta activity and improved the symptoms, right? So they would measure this beta band activity, the local field potentials, and whenever there would come a signal like, okay, it's now happening, it seems to be, the patient seems to suffer from these symptoms, then we can actually reduce this with the stimulus. Okay, so this relation was quite famous. It has been shown also by people from the Charité. So it's a cool new tool, and there will be many things that we can do with it. Okay, so in case deep brain stimulation is not invasive for you, not invasive enough for you, there are many others, right? I told you already about transcranial magnetic stimulation, or short TMS, that was actually approved also for OCD and also already for a long time for major depression. It's such a coil that is placed on top of your head. We'll come to that in a second. And then you will have repetitive sessions with it. So it would be, yeah, how do I say it? You come once a week, maybe you have a session and you come again, but it's totally non-invasive. And there's lesioning, of course, but this has been shown to be harmful in quite many other ways, and it's non-reversible. That's the problem of lesioning. So if you don't get the right lesion, then yeah, no comment. Then there is focused ultrasound, which has been approved as well, FDA approved, and there's deep brain stimulation, which has been approved for essential tremor, Parkinson's disease, and dystonia, as I told you, a special form of OCD already, and most recently, it has been approved for epilepsy. Some forms of epilepsy seem to react well to deep brain stimulation. So everything that has to do with the dynamics of the brain, and especially of the subcortical area, seems to be quite interesting, and there seem to be quite nice effects if you use deep brain stimulation or any other stimulation tools. So just let's talk for a minute or two again about TMS, because what hasn't been done yet is transcranial magnetic stimulation and the virtual brain. I would be also very excited about a project like this, so hopefully you are too, because it's non-invasive, right? It seems to be not harmful. And for example, people with cocaine addiction, they seem to have less relapses after TMS sessions. So same for depression, people seem to feel better after these sessions. And it's totally non-invasive. So in that case, or in that respect, it's much better than DBS. No surgery, no risk-taking there. But it only reaches, and it seems to first only reach the surface, right? So that's why we need surface-based simulations for this. But as we have seen, the stimulus can propagate and maybe also propagate to the subcortical areas. So if you find the right surface piece to stimulate, maybe you find the right target for also changing activity in the subcortical areas. So for TMS, that's it, not been done with TUV before. What has been done has been simulated, I must say, is transcranial direct current stimulation with TVB. So there was an earlier study already from 2016, Kunz et al., and they, in a totally non-data study, so they did it all with the default connector that you know also. They loaded it and they put a model, an oscillator model on it, and then they measured for all the areas the activity, and they also put a monitor, an EEG monitor on top of it, which we have done also in the hands-on session. And then they read after stimulation, so during rest, during stimulation, and they averaged over rest and stimulation, that they looked which of those little networks got activated. So you can see here that during the stimulation, the red ones got activated. And without, in the resting state, there were totally other areas being overly active. So it really changes a lot the dynamics of your brain. If what you can see here by the red things not really overlapping with the green things. So, and the blue things are kind of the overlap. There are some networks being active no matter what, but there are also specific parts of the brain that seem to react strongly only during stimulation. So here, this was in a close relation to what has been published in literature, and it could reproduce a lot of those findings from literature. It hasn't been used here with empirical data yet, so that's now our task to do and include the virtual brain and stimulation with empirical data. And of course, with the virtual brain, there are others using mean field models. And the virtual brain is very user-friendly, and it gives you a very easy access way to implement those mean field models on the brain. But you can also do it, as we have seen in other courses, by yourself and write the code for it and the integrator functions and do it yourself, right? And others have used not the virtual brain, but also whole brain simulations, and they simulated, so the group by Deco et al. Simulated deep brain stimulation for Parkinson's disease as well. They have looked at the whole brain, and they have looked at 10 patients having Parkinson's disease, having a DBS, and they got data from those patients being in the scanner, MRI scanner, with DBS on and off. So here we can see that the different measures that they chose here, let's say all those measures, they were something that could be read by the signal, and they fitted the model, so they could also back this data up with simulation. And then they have that these are healthy controls, age-matched and non-age-matched, so two groups of healthy controls. The patient, when the Parkinson patient, when the DBS was off, was quite far from the healthy controls. Maybe I can even do this. So here we have the healthy controls, and here we have the patient. But once you switch on the electrode, you see that the average was going up slightly. And here it's much more extreme. The patient seems to really be going towards the healthy controls once the DBS is turned on. So they have shown these effects here as well for a third measure that they chose with a whole brain dynamic simulation. So kind of what we already want to do. And they were able to show that the whole dynamic regime was going from from the sick brain towards a healthy regime. So very close in terms of dynamics and bifurcation points and dynamical systems perspective towards the healthy patient. And others have shown that as well. So that seems to be really cool and something we definitely have to reproduce, right? And then there are other studies on mice, which we cannot ignore because we have this, as I already said, track tracing data of mice for the Allen Mouse Brain Atlas. So very detailed data. And they put these mice here in a scanner, and not any scanner, but 9.4 Tesla scanner, right? And then they put electrodes in the mice, deep brain stimulation, and they measured fMRI. And normally there can be, so in the study here by Zenger et al., for example, they had to exclude some regions because of artifacts, because the DBS was actually on and that created some artifact in the scanner. But here they used graphene fiber electrodes that were much more independent of the scanner and didn't create an artifact. And then you can see here, very beautiful, I think, those maps of activation, whether you performed an SDN stimulation of 10 Hertz or 100 Hertz or 30, 250, and how this propagates. So that's really beautiful, I think. So we can, this is very recent study, so we can have much more detailed information now on the data that we are hopeful we can reproduce. And maybe we have to look for mice for that, right? Why not? Okay, and if you are still with me, and I really hope so, then I want to introduce you to the last new bits and pieces of information to you. And yes, this slide for you at home looks a bit different. I wanted to write this, but then with a pen, but then yeah, I will deliver it afterwards, don't worry. A small introduction to control theory. So in case you are a physicist, or maybe you're an engineer, or maybe in your studies somehow, maybe you're a mathematician, then you have maybe heard of control theory. There's this whole great mathematical theory around controlling a system, that it's possible if you control the input to control the output. And for certain systems, this is possible. And especially for man-made systems, which are made by humans, like a power grid or something, when we know exactly all the components of a system, it can be quite nice to study this input-output relationship, and we know everything that's going on in between, and we can really control the system. So controlling the brain is a huge challenge, and a really nice challenge right now in this field of research, because this is actually what we're trying to do with all the stimulation. We are trying to control brain dynamics. We are trying to bring the brain from a certain dynamical state into another dynamical state. So all of this has been written down in equations. And if we are looking at the most simple way of it, we're looking at a linear equation. And here, I hope you can recognize this kind of equation, which looks very similar to the global TVB equation. This is A is a structural matrix, and then X is our state variable, so our variable of interest. That could be, for example, firing rate, right? It's a whole vector then of average firing rates over all regions, let's say. And these firing rates with this structural connectivity are mapped to the next time point, dx dt, right? And then here you have input. So we have seen this, right? We have seen an I, for example, in the TVB equations, yeah? So this hopefully seems a bit familiar. And this is only the linear version of it. And this is, if your system looks a little bit like this, yes, it does for us, plus some noise, right, always. But if we ignore that for a minute, we can say that our network defined by A, is controllable if and only if the controllability Gramian is invertible. So what's the controllability Gramian? That's this nice sum of B and A, where B is your control input matrix and A is your structural connectivity. And you build a sum of it. So this is called controllability Gramian. So this has been shown, that this is exactly if this has been shown. So we can assume that's true. And if our system looks like this, all we have to do to know if the brain is controllable, we just have to compute this. And now, because we all, of course, I know it's non-linear, you know, people will say, oh, it's not a linear system. It's not a linear system, but linear is always the first approximation, and it can get us quite far in many cases. In this case, you're wondering maybe what B is, and B normally is a matrix with all the canonical vectors. So for example, here, this nice matrix with only zeros and ones. Forgive me my not so beautiful handwriting with a pen on the screen here. But this is matrix B, for example. If we have a 3-node system, it's just 1, 0, 0, 0, 1, 0, 0, 0, 1. All the 3 vectors are just ones and zeros. And U would be the strength or your weighting, in our case, our weighting for the stimulus for each individual node. So U would be another vector here, giving us the weighting. Maybe for region 1, we give a stimulus of 0.1, as we have done before. Here, 0.7, there 0.5, who knows? And this would then be our input. So if we assume B is like this, and we can assume it for a linear case and the simplest case, then we don't even need U to know if the system is controllable. We only fill in our B here, our structural connectivity here. We check if this matrix is, we can invert this matrix, and then we have the answer. And of course, this has been done before, not by anybody, but by famous Danielle Basset, my personal hero, the queen of brain network science, I would say. And she is has published that the brain network is actually controllable. And if we fill everything in here, it is controllable, but it's not easy. And why is it not easy? Because, of course, it's very hard to control if you only stimulate or give input to a single node. This is what she found so far. So she used this, and the simplest case here, she used even simpler. So she used B, just simple. I have an eraser, even simpler. So for Baset's case, she said, we will now test every region, as we just did with our hands-on session. We tested for every region whether we can do something to the network, whether we can control the network. So she here tested for every region, whether with a matrix, with a vector B like this, so always the 1 there where we have the region. So B simplifies to a vector, whether then this system is controllable. And this gave her a number, so she can rank for every region. She has done this. She ranked which region is has the highest controllability, they call it, for the network. And this gives us an indication if a region has a high controllability. It means that this region can move the network dynamics from one state to the other state easily. And with that, you can identify regions that could be very interesting for stimulation, right? So another cool goal here is to bring the theory together in practice. And they have recently, the most recent study by the lab in Pennsylvania from Basset, is this study where they looked at the controllability of every region, not for a system in one state, but during a seizure, an epileptic seizure. They made different networks for the different states. So every few seconds, they took together, they made a network and checked which would be here a good target, which region would be would have the highest controllability. Then in the next few seconds, taken together, here this region would be nice, here this region would be best to stimulate and bring the system out of its state, which is in epilepsy during seizure, you want to reach another state than the epileptic seizure state. So quite cool study. I encourage you to read this. There's very simple linear mathematics here behind, as I've just explained to you. And this could be, what could this be used for? For example, if we know which regions are best to be targeted during different phases of a seizure, then we could target them. We could implant a device that does exactly that, stimulates this region at this point and this region where it's reading this, and this talking adaptive DBS. But maybe other forms of direct stimulation could be interesting as well. Often they have electrodes implanted, right? Epileptic patients for 24/7 seizure control management. So lots of cool things happening right now in the field. This was just an introduction to control theory. Just if you are like, okay, there was mathematics, I don't want to listen, okay, then this is a summary slide for you. Controlling the brain is all about bringing brain from this simplified three-dimensional space, from the diseased brain state into the healthy target brain state. And how we want to do it is by using the virtual brain. And one pathway here, pharmacological intervention, for example, can bring, so some medicine can bring the dynamics from the disease state to the healthy state, has been shown here. And this paper you have learned a lot about last time from Leon, where they actually used the virtual brain already to perform an intervention and do like a clinical trial with the virtual brain and show that this is helping to go closer to the healthy target state. For me, myself, I want to do it with DBS, and there are many other measures, TMS and so on, where we can explore if the virtual brain is bringing the dynamical regime closer to a healthy one. Okay, so here, what's the future of brain simulation? We are nearly finished with the lecture here. Yeah, so either it's this, some Neuralink that somebody developed and we all have to wear it, and this is controlling our brain completely, controlling us completely. Or here, a cool view into the future. I like this neuralizer, maybe you know it, right? It's a small device from the movie Men in Black, and it's just erasing all your short-term memory of the Men in Black and the monsters. So it could look like this, but no, this is a reality. The future of brain stimulation, I told you, adaptive DBS is right now a really emerging field of research. People are trying out different strategies. Devices are implanted in real patients, all moving very fast. And then it's moving, as I see it, still towards non-invasive, because an invasive surgery is a lot to take on for a patient. And it's often only performed, if the patient wants to, and it can be scary, of course. So non-invasive seems to be, people seems to respond more openly to that, like a TMS session. So we should definitely aim for non-invasive results. And this modeling assistant, maybe even with real-time computation, maybe even machine learning inside the adaptive DBS device, yeah, scary, can be scary thought, but maybe why not? So this can be, I think this is where we're heading, that depending on where your brain is going, you will just get a stimulus, right? And then you will be right back on it, and you will not have these symptoms that are maybe developing, like bradykinesia, rigidity, and so on. So that would be very cool. Individual level solutions is definitely the way to go. It's the way where we have to be heading, right? In general, personalized medicine, we are not all the same. We have to develop these individual solutions and not assume that one solution fits us all. Same time, I think multimodal monitoring can be very important, because we have some good tools on the level of whole brain monitoring, like the Fmri and so on, but it's not a good spatial resolution. We can get much better spatial resolution from it has not such a good temporal resolution. We can get much better temporal resolution from EEG and MEG. And we should combine this all, because too often people are just looking at one data set and assuming that this is it. And the multi-scale modeling that you've heard about is definitely a trend that is coming up, because people have to make use of all the different data that they have, and they want to put it into one model, because it's all connected. It's all one big network. And then here, just as a comparison on the right side, I just put some printed circuit boards from the 1970s. And now, and what I wanted to show here, maybe you're confused, but my point that I wanted to make here is that this field, for example, of these printed circuit boards has been developing so fast from only a few items here to putting many thousands of items on the smallest space. And then on the other side, we have neuroscience, and neuroscience has not been developing these devices as fast as they possibly could and keeping up with the newest technology. And it's accelerating even more this technology of this PCBs, for example. So probably over the next few years, neuroscience will catch up and we will get even cooler devices, even smaller. For example, the Neuralink device from Elon Musk is chargeable via induction. So you can just put at night some nice head-on and it's charging. And on the lower part here, a tweet from the company Manus TM from virtual reality glove company. So they're making gloves for VR. And here you can also see that it is fascinating. Like 2014, their PCB looked like this, whereas 2 years later only it was tinier than a €1 thing. So there's a lot of developing happening on the technical side as well that we should not ignore and that can be really helpful to us. Okay, so with that, we are done with the lecture part. Sorry that it took me here a few minutes longer, but we have some extra time. We went quite fast through the hands-on. And we will now go on to the second part of the hands-on session, where we will use the graphical user interface in a minute. But first, I want to give some time to questions, comments, anything you want to share about the lecture. Then feel free to do so. So I just hope as a summary that I was able to convey to you that this is a very active field at the moment that holds lots of potential to be developed further, and it will develop very fast over the next few years. And in case you're looking for any cool topic, you know, this could be it. And you get as excited as me about it. So we will watch the stimulus propagate with the TVB graphical user interface now. So for that, I need your full attention with me again. And these are the instructions. We will launch the graphical user interface of TVB. Why will we do that? Because there is a nice way of visualizing a stimulus. And you all want to see a cool video made by yourself of a stimulus propagating. I know you want to. So let's go to ebrains.eu, service the virtual brain. Post it here. Let's go there all together to launch the graphical user interface of TVB. Discard. So, okay. So we have seen on eBrains how we launch the Jupyter Lab, right? But what you maybe haven't seen on eBrains is how to launch the graphical user interface. So ebrains.eu service the virtual brain, hit it. Then you will get on this page of the virtual brain. eBrains, you can also Google it, right? You will find it. And then you go down here to TVB eBrains Cloud Simulator, and you will launch this. And I'm already logged in. But in case you're not logged in, you would have to fill in your eBrains credentials now. Please do so. And then you are in the simulator. This is now the graphical user interface, so you can just click through it, explore it. It's very cool, very easy and user-friendly, I think. And we want to create a simulation, including a stimulus. So first, what you do is you go to project. So can I, for this, just get a yes if you are able to launch and it looks similar to this and you were able to log in and you have in your browser now a graphical user interface of TVB? If you have this, just write a yes in the. Okay, I got a yes, great. So it's possible. Great. Yes, yes, yes. Okay, cool, cool. Thanks for that, guys. Thanks. Cool to see that you're with me. Okay, we will go to project. So maybe you are looking for tabs on top. No, look for tabs on the bottom always when you're using the graphical user interface. Go to project, and then let's check out what the list of all your projects is. And there can be very many projects here if you have worked with it before. Maybe it's also for you the first time to look at it on eBrains, then there's nothing stored yet, right? But in case there are multiple projects, what you need to do is you need to highlight the default project. You need to select this project to work with and make a star in front of it. So if you only have the default project, that's great, there will already be a star. If you have multiple ones, make a star there, make a click. And now, once you highlight this, it means all the other tabs are now related and we will be working with the default project. So now, let's create a stimulus first before we simulate. We want to create a stimulus. So we go down here on stimulus. Hope you found it. Then we can create a region stimulus or a surface stimulus. You can also create a surface stimulus, fine by me, but computationally expensive. Maybe we don't want to crash it today, so let's stay with the region stimulus for now. But feel free later, once there are not 21 people on it, then you can do a surface simulation. I've done it yesterday, and it took about 10 minutes on eBrains, which was already cool and quite fast, but be prepared for that. So this looks already like a pulse train, right? Here on the right side, you can see nicely how the stimulus looks. It goes up, down, it's repetitive. So maybe you remember the T and tau, you have seen the amplitude of the stimulus, that's a new parameter, maybe it's a 1, onset 30, so don't change anything there. I would not change anything there. Just leave it as it is. And we will define the stimulus and we will give it a name. We will not change parameters, that's too much of a hassle, so let's go with the standard parameter. Let's give it a name like stimulus virtual for the virtual course, virtual stimulus. Call it stim virtual, why not? And that's the name. We changed nothing else. I just wrote a name here. So please follow me with it. Just give it any name. So it will look like this. We don't change that, and we will set region scaling on the right side here. So this here defines the pulse train with the parameters, with this connectivity, just as default mode, the default network, and then region scaling. All right, there we can see all regions are getting a stimulus right now. All regions have a check mark. So we don't want all regions. You can do however you like it, but I would deselect them all with this button, right? Here you select them all, here you deselect them all. And we will select, what will we select? Maybe you will see some picture and that will be the stimulus. So I want to select the right primary visual area, but you can select whatever area you like, right? Your favorite area, fine by me. Select an area that you want to stimulate. Give it a scaling. That's the weighting from the code. So maybe something like 0.5. And then you click on set scaling. So I can do this again, click on right primary visual area or any other area that you want. Fill in a scaling. I just took 0.5, you can also take whatever, and click on set scaling. In case you did something wrong, reset scaling, do it again. So this is how you easily in the graphical user interface select an area and a weighting for a stimulus and set the equation. So that's easy, and then we want to save this new stimulus on this region. Ta-da! We saved the stimulus, we made new one, stim_virtual. If many people are now working on it, it can be a bit slower. But don't worry, we'll get there. Have some time left. Okay, now let's see if that was already it. Great. On the right upper side, the operation for creating the stimulus was successfully launched. So hopefully on the upper right side, you will also see something green. And now we want to construct the stimulus. We already selected the connectivity, the default connectivity. And now we want to simulate. So let's launch the simulator with a small rocket here. Simulator. Oh, you see, I have done a lot of simulations already to try it out for us today, but you have maybe none there. So here on the star again, you can just hold your mouse and you already see prepare a new simulation. Yes, this is what we want to do. Click on the star and prepare a new simulation. So now, again, we will just take all the default parameters, so no need to check whether your numbers are aligned with my numbers. Don't worry about that. We will just leave everything default for now. You can play with it later. And now make sure you have here the John Doe 68 connectivity selected. If you have any questions, just reach out on the chat or microphone, right? And everything should be like it is for me as well. We select the conduction suite, a coupling, linear coupling. This is the coupling. Okay, we will say okay to everything. Next, next, next. Here you can select the surface, right? So this at this point, if you want a surface-based simulation, you would, let me go there. You would select here a surface from John Doe, but we want a region-based just for computational usage now. Next, so we can select a spatiotemporal stimulus. Great. So we just created stim virtual. Oh, I created a lot of stim virtuals. Don't know why. Anyway, select, you probably only created one, so it's easier for you. Select those, select the virtual stimulus that you just created. Next, fine, just click next. Again, next, just click next. Next, 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 next. Next, next. Don't change anything. I mean, you can change everything you want, but I'm not changing anything here now. Next, and then we will maybe do something to the simulation length, because I'm afraid this will not work out. So in the last, before you click launch, change the simulation length maybe to 100 milliseconds. That's maybe enough. And then this will be called, the simulation name will be simulation 18 in my case. I guess in your case, it's simulation 1 or something, but it doesn't matter. It would just give automatically a simulation name that you should remember. And we launch. And now you've submitted parameters for simulation length. Please wait for processing step. Great. So now your simulation should be running, same as mine. I can see a lot of stim virtual too. Oh, cool. Then I don't know why. And it's okay, you know, just select one. Bad gateway. That's a new one. I was afraid this could happen. So something like this could happen. It could be that we selected the wrong stim virtual. In that case, you can just create a new simulation, or when you see your simulation here, you click on this here and you copy your simulation. This will copy the simulation. And here is my simulation 18, for example. Everything's still here. And if you want to change it, you should always copy it, or you create just a new one by clicking on the star. So in case Oh, it's green now. Great. The simulation was loaded for you. Okay. Was it launched? Yes. And I see results as well. So I hope you can do that too. If not, try out different things of the Stim Virtual, I'd suggest. We didn't change anything else. So then if you see here on the right side results, just like me, then click on time series region. Time series region. Then we want to see it now. I promised you a video. So then click on visualizers. So on the right side, time series region, then visualizers. And then we want to see it in 3D and 2D. So click on brain activity viewer in 3D and 2D. Let's see if we can launch three-dimensional brain. Yes, great. So you should be able to see already something lighting up, and it's a brain. Yeah, that's already cool. But some things we want to change here. It can look cooler. So follow me for changing. We want to click right side up here. And maybe it's not that intuitive, but if you click up here on this brain, you can change things of the video. For example, let's do some different time steps. Maybe, let's see how fast that is. Maybe 1/4, yeah, because it was very, I think it was very fast. So maybe 1/4 time step different, but feel free to play around with it. Though, then you can choose different color maps, like the TVB color map, for example, color ranges, whatever, however you like it. And then let's also, the only thing I just changed was the speed, upper right side, changing speed. Then select signals from inputs, because here we can see a couple of areas now that are reacting, but we can't see them all. So we select signals from input 1, and we see that only a few areas are selected. I want to see them all, so I click on this all thing. This check mark for all. If you don't want them all, deselect some, so it's okay. And just click next to it to get rid of it again. Okay, so now I also want this to not have a face and not have a navigator, but you can, however you like. But it should look now something like this for you. And we can, if we look from the beginning, and we gave it a visual area stimuli. So it should light up here in the back. First, add okay, and then I don't remember the onset that we gave, but you should get a cool video like this, and you can play around with the onset parameter, with the simulation length. Feel free to do it much longer, of course, and more repetitive stimuli. Then you can see everything interactively, and you get a cool video here that's showing you how the activity is propagating through the whole brain. Of course, we can't see much here right now, because we have chosen default everything and so on, but you can change it. You can change it and adjust your video as much as you'd like to. Load your own brain as connectivity, right? You can do many things, and you can do it all. That's a cool thing on eBrains, with the computational power of the supercomputer behind. So no need to use your local computing power. Okay, so I hope you got a cool video, as I did, to show that you are able to perform stimulations in the virtual brain and can see how that looks. Okay, then we can stop with the virtual brain interface, and I will explain to you the homework already, because yes, we are there already. So for the learning goals, just very quickly, I hope that you agree with me that you've learned a lot today. So for first and for all, you know hopefully now about the choices and advantages when you perform a surface-based simulation with TVB. We have a finer resolution here. We can be more accurate when it comes to the surface and the surface stimulus and so on. Second, you know why and how brain stimulation is performed nowadays. I have shown you some examples, some example studies, and you got hopefully a third learning goal, an overview of the previous studies using TVB and stimulation. I mentioned them all to the best of my knowledge, and also some other literature where people have done similar things. Also, I especially hope the last two goals are reached for you. You are able now to perform your own simulation with TVB. That will be important for the homework. And you're also able to set up and run a surface-based simulation with TVB and GUI. Okay, the GUI thing was rather related to the simulation, but okay. It's interchangeable. I showed you where in the GUI, you just have to load the surface. So feel free to play around with it. I hope that you learned those things and you're confident with it. If not, always feel free to reach out to us, right? Please just use Petra Ritter's email and she will distribute every question you have. If you want to know more, there are all the references. I just want to show them shortly here, where you can read up on everything I told you and find much more detailed information. It's of course way too short, 3 hours, to explain everything about stimulation to you. And another few references, also some videos of our previous talk about where we actually performed this with TVB. So then, before I explain the homework, maybe I explain the homework first, because then we can just have a question session for everything. So for the homework, we will actually do TVB-based, or try out TVB-based therapy. So we will do what hopefully is the future. We will take a patient's brain and it will be sick. And then you will yourself perform stimulation, find out the perfect stimulation in order to cure this patient and make him closer to a healthy patient. I would just explain, it's in the notebook, so I will go to the notebook now. You should send until the next session starts, before February 18th, before the next session starts on February 18th, send your Jupyter notebook to Petra Ritter. She will forward it probably to me, and you will get individual feedback. If you have any other questions on the homework after I explain to you in the notebook, then contact Professor Petra Ritter directly, or use our Slack channel where we can all see and answer your questions. Now let's go back onto the lab eBrains. I hope you didn't close it yet, because all the way in the end of the notebook is homework. So can you cure this patient? So I think that's quite a fun exercise, but it's of course a total toy model. So this is not real patient data. This is not something you would most probably do to cure a patient, but it's going in the same direction. So let's see if we can cure this patient. Imagine that you have to help a patient and his connectivity looks like this. I've just taken the default connectivity and I actually increased all the connections from the right and left primary motor area and doubled their strength, if you want to know what I did, but you don't have to know that. So let's say this patient is suffering from a very rare disease. It's an overload of functional connectivity in the primary motor cortex. So he has too high of a functional connectivity in the primary motor cortex area, because that is what he will have after you just performed a resting state simulation with this connectivity. Now let's assume that you not only have a patient, but a good study always has an age and gender matched healthy control. And the age and gender matched healthy control for this patient, so this is like a mini study, right, is just the default connectivity from TVB, the one with 76 nodes, the one we load when we don't fill anything in. So your task is to help the patient, and he should obtain a functional connectivity around the motor cortex, which is lower than the healthy control. If you find, however, a stimulation strategy that puts this patient back dynamically closer to the healthy control than he is right now, that would be awesome. But it's enough if you just find a stimulation strategy for this patient, which makes his functional connectivity to be lower than the healthy control. So, and to do so, you're not allowed to change back his structural connectivity. That would be a lesion, right? No, we are not touching that. Let's say you are just performing stimulation, but you can directly perform the stimulation on the region as we have seen in the notebook up there. So let's assume you are able to do so. And now feel free to test out different strategies. You can stimulate one region, stimulate multiple, you can use different pulse trains, you can adjust the parameter of the pulse train, and you are only here supposed to use region-based simulations. But if you want to try out a surface-based simulation, feel free to do so as well, however you prefer. And you can stimulate also multiple regions, maybe also with multiple different pulse trains, as you have seen just up one step up here. But first, you should extract the desired measure, and to not get into confusion, what I mean with average of motor cortex activity, functional connectivity, I performed it already and extracted it here for the healthy person. So the desired measure, which we call average functional connectivity, of the right hemispheric primary motor cortex, to be very specific, we will look what is it for the healthy control, because we want to have it lower than the healthy control. So currently it's higher, right? That's something you can guess already. So without any stimulation in resting state, so please use this simulation as your starting point. So it's a very basic simulation, generic 2D oscillator, everything that you have seen so far already. And then I just wrote the data that comes out of the simulation and the time into arrays, because that's easier to handle. And then I extracted something called this functional connectivity matrix, right? And I plotted it. And I just extracted this now. It's not really bold, it's neural activity, but it's just our toy model. So this will be our functional connectivity matrix. That's just a correlation coefficient between the squeezed time series data that we just got from this very simple simulation. And then the measure we are actually interested in, here it's for the healthy person, would be this one. It's the average functional connectivity over all the connections of the right primary motor cortex area. So that's 0.24. Okay, so for you now, I loaded the control, I performed everything for the control. And now you should, next to the patient, load him, perform a simulation without stimulus, check where is he starting in comparison to the healthy control, and then perform different stimulation strategies just by trying out. And it's not hard, it's a very easy and small homework. And make your own notebook for it, an IPython notebook, right? Where you make sure to load everything correctly and so on, and send it to Petra before the next session. So before we are going further, the Kahoot quiz already stopped. Ooh. But let's first finish with this. So just to give you an overview for how it's going further, we have student presentations at the last session on February 18th. And that's presentations from all of your participants. Everybody who wants to present, feel free to present. The presentations are actually required if you want to get the 2 credit points. If you don't want to get the 2 credit points, but you still want to present, you can also just send an email until February 10th to Petra. You have gotten this information via email, and just repeating here, select the topic of your choice. You are very free to do so, whichever topic appeals to you most, and make sure that it contains a demo of your own code, some outline of a concept or hypothesis that you want to test, and a critical discussion. Then Petra will email back and forth with you for modifications of the subject, and you will be assigned a time slot depending on how many people. Then there are the criteria. If you really want this to go in for credits, you should of course, provide sources and cover concisely the topic, answer questions, and so on. And then you need, for the credits, 3 out of 4 homeworks with a pass and a pass in the presentation. I hope this is clear to everybody to get the credits. If you don't want the credits, still feel encouraged just to send an email to Petra for contributing to our last session. Would be really cool. So because you think, oh, there are lots of deadlines, I made this cool overview for you. So in February, make sure before the 10th to send an email and submit the homework of the 3rd session, the one before, about ontologies until February 10th. That's 2 deadlines for February 10th. And then on February 18th, make sure to submit this stimulation homework. And hopefully we see you there for giving, and then will be your turn to give a presentation.